Okay, good morning. We'll go ahead and get started. We brought some glorious British weather for our and I can give you a little more background on that too. The dean that hired me, Bill Stevenson, does anybody remember him? Right. If you remember him, every single talk he ever gave, every presentation he started with British history. So for years, every day we had tidbits of British history. So it's nice to have a British guest with us. It's my pleasure to welcome all of you to the fourth Hugh and Ethel Kelly lecture. We're so glad to have you here, especially David Leigh, who's traveled all the way from England for this special event. And I think you're going to be extremely delighted with some of the things he's going to show. We had a wonderful dinner last night, and I think we all commiserated on the wonderful political environment that we live in, both in America and in Britain. So it's a complicated world, and we had a really wonderful discussion. In general, it's a real honor for ACAS to host this. This is a distinguished lecture series. It commemorates Hugh Kelly's impressive technical accomplishments and the couple's generous support of Virginia Tech. For those of you who don't know, Mr. Kelly earned both his bachelor's and master's in electrical engineering at Virginia Tech. He went on to work at Bell Labs, where he was involved in a number of major projects, and in particular, he helped launch the Telstar communication satellite in 1962. This was a satellite that provided the first transatlantic television feed, which for the United States meant he was featuring Walter Cronkite. Mr. Kelly was a dedicated alum, and after his death, his wife Ethel continued this support as a way of honoring her husband's legacy. When Ethel Kelly passed away, she left a bequest to the College of Engineering, which helped build ICTAS 2 on the other side of campus. And of course, the building we're in today bears their name, Kelly Hall. Her gift also established this lecture series, with the idea being it would bring eminent creative individuals at the top of their fields to speak at Virginia Tech. Today's lecture is also special because we're celebrating 10 years of interdisciplinary research at ICTAS. Indeed, it's a proud time for our institute. Events like this one, where we have visionary thinkers like Leigh, they help to spark conversations about future science and technology. That's really what we're doing here. It embodies ICTAS' goal of enabling ambitious research projects and catalyzing, so to speak, innovation. With that, I want to turn it over to Dr. Harry Gibson, professor in chemistry here, who will tell you a little more about Dr. Leigh.
Thank you, Professor Duma. It's indeed a pleasure for me to introduce David. David received his bachelor's and PhD from the University of Sheffield, and his PhD in 1987, working with Professor Fraser Stoddard, who's well known to all of us in the nano-supramolecular field. And while at Sheffield, David worked on antibiotic chemistry. He then did a postdoc at the National Research Council of Canada in Ottawa, where he worked on conformational analysis, I believe.
Yeah, carbohydrates.
Carbohydrates. I think this background has studied well. As you'll see, he combines both the synthesis and analytical capabilities that are necessary in this field. In 1989, he returned to the UK and had a position at the University of Manchester Institute of Science and Technology, or UNIST, as I believe it's called. And I met him there in 1995. I think that's about the time that he began his work in supramolecular chemistry. Before that, he worked on polymer chemistry for several years. In 1998, he moved from Manchester to Warwick, University of Warwick, and he stayed there for 3 years. And then he was appointed in 2001 as the Forbes Chair in Chemistry at the University of Edinburgh. And he remained there until 2012, when Paul came from Manchester, and he moved back to the University of Manchester. And just last year, he was appointed the Sir Samuel Hall Chair in Chemistry there at Manchester. He's won many awards, and I won't take the time to review all of those, but I'll just name a few. He's a fellow of the Royal Society of Chemistry, a fellow of the Royal Society of Edinburgh. He is a fellow of the Royal Society in Toronto. He's received the International Isaac Christensen Award for Macroscopic Chemistry, the Feynman Prize for Nanotechnology, from the Tilden Prize from the Royal Society of Chemistry. He has published over 400 peer-reviewed papers. It has more than 16,000 citations. His H index is currently 67. Maybe a little bit higher. These things always lag a bit. In my mind, I know several of the people in this field, or in organic and polymer chemistry in general. David is clearly one of the most creative chemists of this era, and I think it's the Kelly committee chose wisely in inviting David to come here. He chooses very challenging targets, as you're going to see, and he pursues those with zeal and good humor. Please join me in welcoming David Leigh.
Thanks very much, Harry, for the kind introduction, and Stefan, and also to ICTAS for the and the Kelly Committee for the very kind invitation to be this year's lecturer, and which is a terrific honour for me, and it's a great opportunity for me to come, and I thought to escape the English weather for a few days, and I visited ICTAS and Virginia Tech, and tell you a little bit about the work that we're doing in a land far away. So what I'm going to talk to you about today actually involves these things, rings, molecular rings. If you pass that around, Harry, because we'll get back to that later, because molecular rings have actually got a very special place in chemistry, but you've got to have the right sort of rings. If those rings are too large, then lots of different things can go in and out of those rings, and you lose some of the benefits of having that cyclic shape and topology. If the rings are too small, then nothing can fit inside those rings, and again, you lose some of the benefits of having this cyclic shape. But if you've got rings that are the right sort of size and shape and chemical functionality, then you can bind things inside those cavities. And if you've chosen the right sort of rings of the right sort of size and the right sort of shape, then if you're very lucky and you wait a long time, then strange things can happen. And what I'm going to be telling you about are the sorts of strange things that you can do with molecular rings, in particular this idea of how to use them to assemble complicated molecular architectures that we think can be used to make things that are sort of like molecular machines. But why do we think it's possible to shrink the concept of a machine to the molecular level anyway? Well, the answer is that there already is a working molecular nanotechnology. It's the one that's used by biology. And the reason that perhaps scientists at the start of the 21st century should be interested in trying to make molecular-sized machines is shown on this slide. Biology uses molecular machines and molecular motors for absolutely every single biological process. So the way that energy is harvested from the sun, the way it's stored in cells, the way that replication occurs, the way that muscles move, the way that you can see what's on this slide and think about it. Every single biological process is due fundamentally to controlled molecular motion and molecular machines. In contrast, mankind at the start of the 21st century, we don't use molecular machines for anything at all. So every material, every polymer, every pharmaceutical, every catalyst, every reagent just relies upon its static or equilibrium properties to have its function. And biology hasn't evolved over 2.5 billion years to use molecular machines for everything for no good reason. And when we learn how to control molecular level motion and how to interface those sorts of motions with other molecules in the outside world, then I'm convinced it will change our whole approach to functional material and molecular design. Now, how are we going to do that? Well, it's not going to be by shrinking engineering concepts down from the big world to the very small, because as you know, the way that matter behaves at different length scales is just so different. So this bottle of water, which is in the big world, is hopefully just going to stay rooted to the spot on that table during my talk, unless I happen to go past and give it some kinetic energy and send it sprawling. But if this was a molecular-sized object, it wouldn't be stationary at all. It would be constantly moving through random thermal motion, through Brownian motion. And that just illustrates one of the differences between how matter behaves at the molecular scale and in the big world. And if we're going to try and make machines that operate at the molecular level, the nanoscale, then we're going to have to use engineering concepts that are appropriate to that length scale. And to do that, it seems sensible to learn lessons from how biology copes with making molecular machines that operate in that environment, and to use those sorts of properties and characteristics to design synthetic sort of machines. And these are just some of the sort of characteristics of biological machines that it seems sensible to try and apply to synthetic systems. And in particular, I hope to convince you of these three things at the bottom. First of all, that concepts such as momentum and inertia are irrelevant at the nanoscale. And so molecular machines are not going to operate through Newtonian mechanics. They have to use statistical mechanisms in order to operate. So you have to design your systems accordingly. Secondly, we go back a slide, we can see that all these biological machines either walk along axes, move along axes, or they rotate around, move along tracks, or they rotate around axes. And this is for a particular reason, that doing so enables the machine only to have to worry about this controlling movement in a particular dimension. So these architectures are used to restrict the degrees of freedom of the components to only the one that the machine wants to operate in. And finally, most importantly of all, what biology really uses molecular machines for, and molecular motors, is to operate molecular systems away from equilibrium. So we've had 40 years of supramolecular chemistry, which has all been about doing chemistry under thermodynamic control and moving towards thermodynamic equilibrium. But at equilibrium, there is no function. Nothing useful can be done. And what biology uses molecular motors and machines for is to shift chemical systems and molecular systems away from equilibrium where they can have function. And I'm convinced that the next 10, 20 years are going to be all about how to do that with molecular systems. So what are the architectures that we're going to use? Well, they're shown by these sophisticated molecular models, not very stable. So this is one of the sort of architectures that we're going to see, where we have two or more molecular rings that are linked together. They're not connected by any chemical bonds, but they can't be broken apart without breaking a chemical bond. And these sorts of molecules are called catenanes, from the Latin catena, meaning chain, because as you can see, they're like links of a chain. And the only interesting thing about catenanes, what makes them different to other sorts of molecular architectures, is that the components can undergo these large amplitude motions, one through the other. And if you control that rotational motion, perhaps you can use that as the basis of a molecular machine. The second sort of architectures that we're going to see is when one of those rings has been replaced by a linear unit, and with bulky groups on the end, so that the ring can't fall off the track. And these sorts of molecules are called rotaxanes, again from the Latin rota, meaning wheel, and axis, meaning axis. And, the nice thing about rotaxanes is that the ring can sit on different positions on the track, and if you can move it, say, from the yellow part to the white part, then that's controlling translational motion. If you can control translational motion and rotational motion, then that's everything. But how are we going to make these sort of structures? It's hard enough to thread the eye of a needle in the big world. How can we thread the eye of a needle when that eye of the needle is only 1 nanometer in diameter? Well, the answer again is to use some sort of concept from biology, and in particular this one of self-assembly. Well, I was trying to think how to illustrate the concept of self-assembly to you. all today. And I got up this morning to go and get the newspaper, and there was a little dog had got its way into the inn at Virginia Tech and had made a right mess of the newspaper, which was of course a bit of a disaster for me. But it did get me thinking that of course a newspaper is a very complicated, information-rich species which requires lots of effort from lots of different people to put together every day. You've got to have the writers to write the articles. You've got to have the editors, the copy editors, to edit what's written and put it together onto the page so that it all fits. It's all the right number of words and the right number of articles. You've then got to have it sort of copied to set and then sent out to the printers to actually print the newspaper. And finally, of course, it's got to be delivered to all of us so that we can all get to actually read the newspaper. So it's a very complicated process to produce this information-rich structure. And I wonder, wouldn't it be easier if there was a way of just sort of taking the building blocks and getting them to sort of assemble together to produce the actual structure that we want. And so that's the sort of thing that we try and do in our group. We try and get the molecules to do the hard work and build themselves, often using non-covalent interactions such as hydrogen bonding to put them all together. And in fact, we got into this area by chance, not deliberately at all. This was about 20 years ago now, when we were trying to do something completely different. We were trying to make cyclic structures such as this, which we hoped would bind to carbon dioxide, to maybe be able to scrub carbon dioxide from the atmosphere. And this molecular ring is the right sort of size and shape to bind to carbon dioxide. And the idea is that these hydrogen bonding groups, these are hydrogen atoms, the idea is that these would hydrogen bond to the oxygen atoms of CO2 in the cavity and perhaps be able to scrub the CO2 from the atmosphere. However, when we tried to build this molecule by taking isophenol dichloride and xylyldiamine and mixing them together in a non-polar solvent, we got a surprise because we didn't get a single ring at all. What we actually got were two of these rings interlocked together, fed one through the other to form a catenary. And it's perhaps easier to see from the X-ray crystal structure where this is a technique that gives the precise positions of all atoms in a molecule. And you can see that I've drawn one of the carbon frameworks of one ring in yellow and the other in light blue. And you can see that they really are interlocked, one threaded through the other, without being connected together by any chemical bonds. What there is between the two rings are these hydrogen bonds shown with the dotted lines between the amide groups of one ring and the amide groups of the other. And these 6 hydrogen bonds are what actually template the formation of the structure. If you remember, these amide groups were designed to bind to the oxygen atom of CO2, but what actually happens in this chemical reaction is once one of the rings is formed, these hydrogen bonding interactions bind to an oxygen atom on another fragment, thread that through the cavity, and then that reacts further to form the cyclic structure, so templating this formation of the catenary. So this is a one-step, 8-molecule condensation, 4 of these and 4 of these, to form a mechanically interlocked structure. And it was, when it was discovered 20 years ago, and still is today, probably the simplest way of forming threaded molecular structures that's known. So this was a surprising result. So just a serendipitous discovery in the lab. But surprising things happen all the time. And it's not just having something unexpected occur in the lab that's the important thing, of course. It's then what you can do with it. And we recognise that we had this very efficient way of forming these mechanically interlocked structures that would allow large amplitude motions. We wondered, was it possible to use these sort of structures to control the motion of the components and actually use that perhaps as the basis of some sort of molecular machine. And so I'm going to sort of tell you So how we do that with the last sort of 30 minutes or so. So it turns out you can make not just catenanes in this way, but also rotaxanes. So what's happened to the metal ring? Does someone have that? The metal ring that we pass around to the top. Oh, could you come and give me a hand? Would you mind? Fantastic. Yes, a couple of things there. Very good. Thanks very much. So your name is Chantal?
Yes.
Nice to meet you. I don't want to touch the room, but if you come here, if you just come here, so that everyone can see. So you're a chemist? A biochemist. This will be easy for you then. This is going to be really straightforward. So we're going to try and make a rotaxane molecule. So if you could hold the ring up here, then what we have, just twist it, that's perfect. So what we have here is we have a ring, we have a thread, we have two stoppers, one somewhat bulkier than the other, I have to say. So what we'll do is I'll put that here, and then I just need to add a little bit of catalyst on there. And okay, Chantal, if you just drop the ring, just drop it. We've got this on. Well done, Chantal. Well done. Quantitative yield. That's great. Well done, Chantal. So what happened there was we have the ring, and on the thread we have these hydrogen bonding groups which are complementary to the ones on the ring. When we add the little catalyst that I sprinkled on the top, this opens and closes these double bonds many times, allows the rings to thread on, and as you saw, you can get 100% yield of these interlock products in this kind of way. So it's really possible to form these sorts of structures. And in fact, it turns out you can use lots of other kind of templating groups to assemble these sorts of interlocked systems to form the structures. Some of them are particularly high yielding, but there's lots of different sorts of functionalities of chemical groups that we can use to form these kind of coded structures. What can we do with them? Perhaps bring the lights down a bit for these ones? Yeah, that's what okay, just for these ones. So this is the example of the sort of things you can do with these molecules. This is a rotaxane of the sort that we just showed, that Chantal made. This is, it has two different binding sites on the axle here. One of them is this fumaramide group, so that's two amide groups separated by a trans carbon-carbon double bond. And this is a very rigid system that holds these oxygen atoms in the perfect position to form hydrogen bonds with the amide groups of this macrocycle. So normally this is the best site, the preferred site for the macrocycle, the ring to reside on the axle. However, we can do chemistry on this molecule once we've made it, and if we shine light on the carbon-carbon double bond, UV light, we can isomerize it, change its shape from trans to cis, as is shown here. And in this state, the molecule, this region is largely self-satisfying in terms of hydrogen bonding, and so the macrocycle the ring moves down to the other side of the track and binds on this other brown station or binding site, which is not as good a binder as this one, but it's better than that one. And this binding site has got fluorocarbon units between the amide groups, and these fluorocarbon groups are like the constituents of Teflon. So this Teflon-like group can either be exposed to the outside world or concealed from the outside world just by shining UV light on this molecule. And the other feature that we've got of this molecule is that we've got pyridine groups, nitrogen atoms on the ends of the macrocycle. And this is so that we can attach these molecules to a surface. So this is a gold 111 surface, a very flat gold surface, to which we add alkyl chain thiol carboxylic acids. And these form a very well-known type of self-assembled monolayer, so just one layer thick of these thiol alkyl chain carboxylic acids on the gold surface. And if we expose that material to a solution of the rotaxane, then the pyridine groups of the macrocycle of the rotaxane will form salt bridges with these carboxylic acid groups, and we get a monolayer, just one layer thick of these rotaxane molecules on the surface. And if we start with the trans double bond rotaxane here, then the rings are all over this green station, And all these fluorocarbon units, all these Teflon-like groups are exposed to the outside world, and so the surface is relatively hydrophobic. But if we shine UV light on this surface, then that will cause this isomerization reaction, the change of shape of this group to occur, and that will cause shuffling of the axle up and down the surface, and the fluorocarbon groups, the Teflon-like groups, will get covered up by the rings, and the surface properties should change. And this now is a picture and videos of that material in action. So this is the gold surface with the monolayer, so just one layer thick of these rotaxane molecules on the surface, obviously too small to see, with all the fluorocarbon units, the Teflon-like groups exposed. And now on top of that, we've added a tiny droplet, a microliter droplet, you'll see just over 1 millimeter in diameter, of diadomethane onto that. It has to be a high boiling point liquid, because this has got a very large surface area to volume ratio, and so water, for example, would just immediately evaporate with a droplet of this size. So you can see there's quite a large contact angle here of the droplet on the surface because it doesn't like the Teflon-like groups. And now what we're going to do is irradiate with UV light, not over the whole surface, but just to the front of the droplet. And this is a video of what happens when you do that. So here's the droplet on the surface, the gold surface, with the one layer thick of the Teflon coated rotaxanes on this surface, a high contact angle, and now we're going to irradiate with UV light here, and so in this region, the fluorocarbon units are going to get covered up in this region where the light shines on it. So we've just started irradiating now in this region here. So in this region, the Teflon-like groups are starting to get covered up by the rings, and after a certain amount of time, you can see the droplet starts to wet that part of the surface because it likes it better. And after a little bit longer, even the rear of the droplet starts to move. So the whole of this droplet that you can see with your naked eye is being transported across this surface just by the action of light on a monolayer, one layer thick, of these synthetic, very simple molecular machines. And when the motion is finished, you can see that there's been a contact angle change because the fluorocarbon units in this part have been covered up. And if you do exactly the same experiment but without fluorine atoms in the molecule, you see no change in droplet shape and no movement of the droplet. So the movement is really down, the droplet is really down to the concealing and revealing of these fluorocarbon groups. You can even use this material to do work. This is a 1 in 8 slope, which is probably more than I can do on a bicycle. But if you put this droplet on this 1 in 8 slope of a monolayer of rotaxane molecules and shine UV light to the front edge of the droplet, you can actually get it to climb uphill against the force of gravity just by the action of light on this monolayer of one layer thick of molecular machines. So that's the sort of thing you can do with these rotaxane molecules, these simple molecular machines where you're just changing the positions of the rings between two different sites on the track. But we've been interested for the last few years in how we can make much more complicated sort of machines, much more like the amazing molecular machines that are found in biology. We've wondered how can we make repetitively acting machines, things that progressively do more and more work as you fuel them. How do we drive molecular systems, chemical systems away from equilibrium? Really, how do we make motors? And this is a question that has was known when we started this work, but really wasn't known to chemists. But these ideas come from solutions to non-equilibrium statistical physics problems. And in fact, this problem of how to take Brownian motion components, so the components of molecules are all moving just by Brownian motion, so that how to get directional Brownian motion from Brownian particles has vexed physicists for 150 years, in fact, dating back to really the origins of the laws of thermodynamics. And James Clerk Maxwell, the Scottish physicist who, along with Boltzmann, discovered the kinetic theory of heat, was one of the physicists who was involved in formulating the laws of thermodynamics in the 1850s and 60s, and he realised that there was a paradox between what he ultimately come up with for the theory of heat and the laws of thermodynamics. And he illustrated this paradox by way of a thought experiment that's come to be known as Maxwell's beam. And that thought experiment is this. Imagine a box that contains gas molecules which are, some of which are hotter than average, so faster moving than average, those are shown in red, and some of which are slower moving than average, so they're cold, so they're shown in blue. And imagine there's a partition in this box operated by some being that's come to be known as Maxwell's demon. What this being does is there's a trap door in this partition, and the being can open it when it sees a hot or a fast moving particle approaching from the left, or a slow moving particle, cold particle, moving from the right. Then the being will open the door to let it through. But he won't let it through, or he won't let it through, let slow molecules through from left to right, or fast molecules from right to left. So if the being does this, it should be able to sort the molecules into slow-moving ones or cold ones on one side, and fast-moving particles or hot ones on the other side. That would set up, of course, a temperature gradient between the two sides. And if this is a frictionless door, then it looks like the being spends no energy to do this sorting operation because they're just moving by Brownian motion. And yet you can set up a temperature gradient, which of course you could use to drive the turbine and get work out of it. So this would break the second law of thermodynamics. You would be able to get work out of a system by putting no work in. It would be a perpetual motion machine. You would get an endless source of energy from nothing. And Maxwell realised, in fact, that there were other sorts of demons, not just temperature demons, the beings that sorted particles according to temperature, but he realised that there were other ones where you could even just sort things by way of position. If you had a trapdoor here and the being opened it just to let things through from the left to the right, but not from the right to the left, then that would establish ultimately a pressure gradient between the two sides, which again could be used to drive a turbine and get energy out. So Maxwell and other physicists realised that this was a paradox, something was wrong, but they didn't know what that was. And it took nearly 100 years for actually physicists to finally sort out the details of what the solution to this paradox is. And of course what it is that the being, to know when to open the gate to let the particles through or to close the gate, has to process information to know when to do that. And that processing of information always costs more energy than you can get out at the end. So you cannot use this to break the second law of thermodynamics and solve the world's energy crisis. And it's of course why your computers get hot when they're working and so on. But from our point of view of trying to make molecular motors, this is very interesting because we've gone to systems that are out of equilibrium, from systems that were originally at equilibrium, so this was the original system at equilibrium, and just by this processing of information, we've gone to systems that are out of equilibrium, and that's exactly what a motor does, a molecular motor. And so we wondered, is it possible to make to use this as the basis of making a molecular motor? Not to try and break the second law of thermodynamics, we're going to put in a lot more energy than we get out, but in order to drive the system away from equilibrium. And the way that we do that is actually to get molecules that process information, that they read, that we give them instructions and we read back signals from these species. So I want to show you how that kind of works with a molecular system. So I'd like need a volunteer to help me with this. Yes, would you mind, you at the end, if you would mind coming out to just give me a hand with this, if that's okay? We'll try and get this done. So if you could just come over here, that would be great. So your name is
Sophia.
Sophia. Okay, if you just stand here, that's brilliant. So Sophia, we're going to play a little game with cards. So are you familiar with cards? You play a lot of poker online, and so you know all the different suits. So hearts, clubs, diamonds, spades, and you know the different cards, so queens, aces, and so on. I always have to go through these in case sometimes people aren't familiar with the various kinds. So queens, let me find some other on the cards, just to show you it's an all package. So jacks, you're familiar with those, and kings. Yes, okay. So what we're going to do is we're going to play a little game where I'm going to turn my back. I want you to just go through the cards, choose any one that you like, look at it, so you know what it is, and then put the rest of the cards down and hold it like this so that I can't see above or below. Is that okay? So you can flip through, you can look through the front or the back. Okay? So whichever you like. Have you got one?
I do.
Okay, so you put the other cards back on the table? So
Yeah.
okay, great. Okay, so we're going to play a little game where I'm going to try and figure out hopefully what the card is. So first of all, are you married, boyfriend, girlfriend? No, single. Okay, yeah, that's a good good. Okay. So, great. So is it okay if I hold your hands? Yes. It's got nothing to do with the trick, but I won't do that. I'll just try and do it. So what we're going to do, we're going to play a little game. I'm going to ask you questions. I don't want you to give anything away in terms of signals. So I want you to either be blank, not give it, or lie to me with your eyes. Lie, try to be deceitful, pretend I'm your boyfriend, that sort of thing. Okay. All right, so I'm going to ask you, let's give it a feel. So it's either a black card or a red card. Okay, it's a black card. It's a club, it's a spade. Okay, it's a red card. Don't help. It's a heart, it's a diamond. Okay, it's a heart, it's a high heart, it's a low heart. It's a diamond. Don't help. It's a diamond. It's a high diamond, a low diamond, middle diamond. It's 9, 8, 7, 6, 5, 6, 7, 8. I think you should show everyone the 7 of diamonds. Well, there you go. There you go. Great job. Good job. So we're all made of molecules. If you press the right buttons and right sort of interrogation, you can read back the signals and get the information. Oh, this is my wife and daughter. My daughter's there to remind me that not everyone is as well behaved as audience participants, and my wife's there to remind me that I can't read everyone's signals. So, so this is the Maxwell demon experiment. How can we mimic that with a molecular species? So this is a molecule that mimics exactly the Maxwell demon thought experiment, but in a non-diabetic sense. So we're going to put in a lot more energy than we get out, but we're going to drive the system away from equilibrium. So we have 2 compartments separated by a barrier. And what we need is that the Brownian particles, these are the rings that just undergo Brownian motion, we need to, in order to drive it away from equilibrium, we want to have a system where when the ring approaches the barrier from one side, from the left, the barrier will go down and the demon will let it through. However, if the ring approaches from the right, the demon won't let it. There'll be a higher barrier. And then if you can do that, you can drive the distribution of the rings away from equilibrium without ever changing the affinity of the rings for the 2 different groups, and just drive the thing away from equilibrium. And this is what a theoretical physicist would call an information ratchet mechanism, a type of Brownian ratchet mechanism. And this is a molecule that does exactly that. So it has, it's a rotaxane. This one isn't based on amide groups, it's based on these sorts of molecules which are called crown ethers, and ammonium ions on the binding sites on the track. So it has 2 binding sites on the track, and a barrier between them, which is this still-being group, which is a double bond with 2 phenyl groups on either side. If the double bond has this sort of shape, then the ring can get through to the other, between the 2 sides, and so the gate is open, if you like. If this is isomerized, changes shape, so that the phenyl group here, this group is on the same side as the other one, then the movement of this ring is blocked. and it can't get through, so the gate is closed. Then on the macrocycle here, on the ring, we have this group here, which is called benzophenone, and this is what's called a photosensitizer. So if you shine UV light on this molecule, this absorbs energy, and it can transfer it to this gate functional group and cause it to change shape. So it can open the gate or close the gate behind the molecule. But it can only do this when it's very close in space. And this binding site is very close to the gate, but this binding site is very far from the gate. So if this ring is over here, and the photon is absorbed by the ring when it's over here, then that will just be lost as heat, and it won't transfer the energy to the door to open or close the gate. Whereas if it's here, if it's on this binding site and it absorbs the photon, it can open the gate. These purple triangles, purple rhombuses or diamonds, show the position of the ring, the amount of ring on the left-hand side. The amount of gate that's open. So we start off with 100% of the gate that's open, and eventually we'll go down with that. And the blue squares show the amount of rings that are on the left-hand side. So with 100% of the gates open, so the ring is free to move between the two sides, we see that there are 65% of the rings are on this side. So the equilibrium position is actually 65% of the rings here, 35% of the rings on the other side. And if we shine UV light on the molecule, then we start to isomerize this double bond, change shape of this double bond, so we get more and more of this closing until we get down to a steady state of about 40% of these are open and 60% of them closed. But you'll see the amount of rings on this side is still 65%, it's still the equilibrium value. This is because the information ratchet mechanism can't operate without a mechanism for closing the gate, closing the door, once this ring has moved to the other side. And so to introduce that, we add an external photosensitizer as well. This is called benzyl. And this will close the gate irrespective of the position of the ring on the track. It doesn't mind whether the ring's here or the ring's there. So it can close the gate even when this has gone through to the other side. And once we add benzyl, the mechanism for closing the gate, and we carry on irradiating, you can see that now the amount of rings on the left-hand side starts to go down until the majority of the rings is actually on the other side, even though we've never changed the affinity of the ring for the two binding sites, and we've driven it away from equilibrium. So this is, in fact, an information ratchet mechanism and a molecular motor mechanism. But, and that was sort of the first synthetic system to be made that did this sort of motor mechanism. But many of the biological motors don't use light to drive them. They use chemical fuels. So, for example, motor proteins typically catalyze a molecule called ATP in the body in order to do work. So if we're going to try and mimic some of these amazing biological molecular machines, it seems that we should try and make a system that can use a chemical fuel just like they do. And earlier this year we reported a rotary molecular motor which does just that. So how this works is that it's a catenane with a ring, a small ring on a bigger track with bulky groups attached to it. And what happens is that as fuel is added to this mixture, these bulky groups come on and off, and they're able to actually drive the ring directionally round. This action is able to drive the rings directionally round this track. The way that it works is it's very similar to the Maxwell-Demon-Rowe taxane that we saw before, but this is cyclic, so it's just like sort of taking this system and cyclising it round so that when the rings get on that side, they then get driven back to the start to make this move directionally. And instead of using the light to cause this chemical isomerization reaction in the previous one, what we have here are bulky groups come on and off by chemical reactions to cause the ring to move directionally. And this is autonomous, so it rotates directionally as long as the chemical fuel is present. So it's just like having your car, that if you put gas into the tank, then the ring will spin directionally, and then as soon as all the gas is used up, then it will stop. Now the mechanism, explaining how this mechanism works is a little bit complicated. It basically requires the positions of these rings to determine how fast these the blocking groups come in and out of place. But in this particular case, the referee, one of the referees of the paper, came up with a good analogy of explaining how this works. So this is a famous British cartoon. I'm not sure if you know about this guy, these guys in the States. This is called Wallace and Gromit. The dog is called Gromit, and the guy at the back is called Wallace. And this is one of their cartoons called the Wrong Trousers. And in this, Wallace and Gromit are on this train track. The track runs out, and Gromit puts down track in front of the train so it can keep going. And this is like the situation that we have here. The reason that this works is that the train, if you like, is constantly moving backwards and forwards, but there's only track in front of the train. What's the difference between our system and this is that, as the referee says, that here the track is being removed from behind in our molecular machine to make this work. So we like this analogy so much that we had this picture made for us of where we have Gromit putting down track, and that corresponds to the blocking groups being removed. That allows the train to move forward. And Wallace is picking up track from behind the train so it can't go backwards. That corresponds to the blocking groups going down. And we made a short little movie to illustrate that. If I get that in the track, then Wallace moves so it can't go backwards. And Gromit puts down the it's on for an hour and a half. No. So this really works. I don't want to go into the details of it, but these are experiments that shows as the fuel is added, the ring moves directionally around a bigger one. And that's shown by these sort of experiments showing that this really works. And this is the first example of an autonomous, chemically fueled, synthetic molecular motor. So that's, I say we're sort of really inspired by these astonishing biological machines. And this is the most amazing molecular machine of all. This is the ribosome. So this is an astonishing molecular machine. It's in every one of your cells, in every one of any living cell. And it's what converts genetic information in the form of a messenger RNA strand, which has been derived from DNA. Then the codons on the strands here tell the ribosome which amino acid building block to add next to a growing polymer chain. And this is how the ribosome puts those together in the precise order dictated by these codons of the messenger RNA, and what comes out is a protein with a specific sequence of amino acids that then folds to form your protein. So the ribosome is an extraordinary molecular machine. It's got a molecular weight of between 2.8 and 4.5 million daltons. So it contains about 1 million atoms in this molecular machine. It's made mainly from RNA. It makes between 15 and 20 amide bonds a second. And just the discovery of the ribosome led to the award of the Nobel Prize for Physiology and Medicine in 1974. And 7 years ago, the Nobel Prize for Chemistry was given to Venki Ramakrishnan, Tom Stites, and Avi Yonath for their structural elucidation and mechanism of the ribosome from X-ray crystallography and other sorts of methods. So how can we possibly make a synthetic molecular machine that's anything like the ribosome, anything even remotely like it? Well, this is a rotaxane molecule which does a tiny, tiny part of what the ribosome does. It consists of a track with 3 building blocks on it, several building blocks on it here, a ring which is threaded on the track by, in this case, by a catalytic chemical reaction involving a metal ion, and a bulky group that holds the ring on the track from the other end. So what actually happens is this ring binds to a metal ion, that catalyzes a chemical reaction through the centre of the ring to assemble the ring onto the track. We then add a sort of robotic arm and then operate the machine. And then this ring can move down the track, picking off the building blocks in the order that they appear on the track, transferring this to a new site, regenerating the catalytic arm so it can go and pick up the next one, and so on, as this moves down the track, and it's building up a new sequence polymer using the information from the track. And this is the design of that molecule. So it's a rotaxane architecture in order to confer precocity. So this ring can't get off it can't go past any of these building blocks until it's picked them off the track. So the architecture stops this from being able to react out of sequence. It can only react with first the green one, then the purple one, then the red one. The bulky stopper on this side actually provides directionality. So this ring actually is going to move randomly up and down the track, but any time it goes backwards, it just bangs into this bulky group and doesn't react with anything, so it doesn't matter. But the only productive reactions are when it moves from left to right. So this provides directionality to the system. These barriers are acyl amino acid building blocks, and the sequence they appear on this track is the sequence that they're going to be built into the new polymer. And the macrocycle here contains a catalytic arm, 2 sides, a catalytic arm which is going to pick up the building blocks, and then an elongation site where the building blocks that are picked off are passed to, so that where the new polymer will grow, and that regenerates the catalytic arm. And the chemistry that's used for that is native chemical ligation, which is, so this reactive arm is actually a peptide. It contains a cysteine unit which contains a thiol group, a sulfur atom, and this sulfur atom is able to react with the stoppers, which, as I said, were acyl amino acid derivatives, so they're esters of amino acids, and esters will react with thiols to form the thioester. And so this is able to pluck off the groups from the track here to form the thioester. Once the thioester has formed, so the acyl amino acid groups being removed from the track, then the ring can move over this, but it can't move any further until this catalytic arm is regenerated so that it can attack the next building block. And this is regenerated by passing the building block to this elongation site to a cyclic transition state where this amine comes around and pulls the acyl group off the thioester to form an amide bond here. And that regenerates the thiol while simultaneously adding the building block to the elongation site. And now this can react with the next group along to another native chemical ligation reaction, and so on. We have rigid spaces between these building blocks to prevent folding and reaction out of sequence. And this one is made not through hydrogen bonding, which the previous rotaxanes and catanes I've talked about have been, but using, we don't want to have strong interactions between the ring and the track in this case, because we want the ring to be free to move up and down the track, just restricted by these building blocks. So we use the metal catalyzed reaction to assemble it, because there are no residual intercomponent interactions left after that reaction. So this is the chemical structure of the molecule that corresponds to this cartoon. We make it with acid labile protecting groups in place. This makes the machine completely stable in this form. It can be treated just like a normal chemical structure. And to operate it, we just remove these protecting groups, these locking groups with acid, and then put it into a basic buffer, leave it for 36 hours at 60 degrees C, and when we come back, there's just 2 components left in solution. There's a track which no longer contains any amino acid building blocks, and there's a ring which has got a peptide with a new building blocks added to it, and all the characterization data shows this molecule to be identical to an authentic sample that's been synthesized by conventional peptide synthesis. So it really does have this sequence. But in fact, we can actually sequence this directly, even the operation product by mass spectrometry. So we can use a technique called MSMS, tandem mass spectrometry, to sequence the molecular ion that is actually formed. And when we do that, then we take some derivative, look at the MSMS spectra, and these fragmentation patterns here correspond to the loss of exactly the amino acid groups that we would expect, the alanine, the leucine, the phenyl alanine group, and nothing else is formed here. So the product that we get is exactly the sequence that of the building blocks were originally on the track. So we're very excited by this idea. We hope to use it to make other sorts of peptide derivatives, helices, beta terms, beta sheets, and so on. But perhaps most excitingly, it would be really cool if we could use this to control the sequence of other types of reactions of building blocks, which aren't necessarily those ones that are found in biology. So sequence at the molecular level turns out to be extremely important for properties. So if we just consider proteins, for example, proteins are all made from just 20 odd naturally occurring amino acid building blocks, and yet the sequence of those building blocks, which ones are incorporated, can lead to all the incredible variety of properties that different proteins have. So for example, one sequence of amino acids gives you copropenin, which is has been used as the basis of looking for drugs for ACE inhibitors for heart disease. Change the sequence and you get myosin, which is a protein that is responsible for the movement of muscles. So you can make something that can actually generate force. Change the sequence of building blocks and you get spidersil, something that's twice as strong as steel. Change the sequence, and so on and so forth. And yet all the biology is just based on just 4 different types of sequence polymers: proteins, DNA, RNA, and carbohydrates. But we have the whole of the periodic table and the whole of 150 years of chemistry to be able to play with different building blocks and different sorts of chemical reactions. Perhaps we can do something that's currently impossible to do with conventional polymer chemistry, and that's to make polymers that have different sequences. So maybe something like polystyrene that can have derivatives fold in exactly the same way as proteins do, to get lots of different molecular materials with different properties. So just as biology makes sequence polymers with molecular machines, perhaps one day we will be able to use synthetic molecular machines to make other kinds of sequence polymers in sort of molecular factories, just in the same way as cars and other objects are made today. That was, we reported the first example of this a few years ago, 3 years ago now. Subsequently, we found out that it's easier to actually form the rotaxane first and then click on to the a little rotaxane monomer, whatever polymer chain that you want on the end here. And if we do that, we can actually go to using, this is a polystyrene derivative made by atom transfer radical polymerization, which has is copolymerized with a styrene residue that's got an amino acid attached to it, leucine. And if we operate this molecular machine on this sort of track, then the information that's transferred by this operation isn't the sequence of the building blocks, because these are all leucine, but it's the distribution of the leucine's molecular weight between the polymer strands that's transferred to this new polymer that's produced by the ring moving down this track. And this is just showing mass spectrometry, showing that we can, that is indeed what's transferred. The information from this polymer is transferred into the resulting one. And we've chosen leucine for a particular reason here, and that's because if we make leucine monomers of with this sort of polymer, we can make systems that will fold up as polyleucine does, to form an alpha helix. And this helix has chiral information associated with it and can act as a catalyst. And so the material that we make from this molecular machine is able to act as a catalyst. This is just showing it has the alpha helix. And this will catalyze the epoxidation of a chemical reaction of this chalcone derivative. And you can form in very good yield and very good in antimerric excess just one-handedness of this epoxide by using this asymmetric catalyst. So this is a molecular machine, synthetic molecular machine, that picks up building blocks by going down the track to make a new polymer that then folds to have a tertiary structure which is able to asymmetrically catalyze a chemical reaction. So that's the state of play that we've got to at the moment. I've just one last thing I wanted to show you before I went. This is a slide that I showed earlier showing that lots of different groups can be used to form these rotaxane molecules. I was really excited to be coming here to Virginia Tech today and telling you all about supramolecular chemistry. And this slide sort of got me thinking that really supramolecular chemistry, it's a lot like love. You know, there's a special one for all of us, but sometimes almost anything will do. And I want to illustrate that with you today. So I'm volunteer. Sorry, Chris, would you mind coming and give me a hand for this, if you really mind? Chris picked me up from the hotel today and said that she wanted to do some magic with she didn't say that, but she did pick me up today. Chris, if you could stand here, we've got one last little one to do. So Chris, it was only a short 30 seconds in the car today, but are you married or boyfriend? Married? Oh, that's a shame. But we're going to try and give you a chance to trade up. So, husband's not in the audience. Okay, that's fantastic. So, what we're going to do, as I say, supramolecular chemistry is a lot like that, but we've got to, it's a random process. So, first of all, you need to find what your sort of type is. So, this is Harry Styles from One Direction. Is he your type or not so much? No, okay. What about, Prince Harry? A bit of royalty? No, still not. Oh, what about George Clooney? No, still, oh gosh. Oh, my own favourite, Super David Beckham, former what about James Bond, Daniel Craig? No, none of them. Sorry, hard lady to please. We'll try and find out anyway which one of those might be the best for you. So what you need to do is to take the cards, and then you're going to do, you're going to say he loves me, and goes to the bottom. And then he loves me not down here. Then he loves me not. He loves me not. He loves me to the bottom. He loves me not, throw away. He loves me to the bottom. He loves me not. So for me, it would be Super David Beckham. Okay, but we'll tell you. So we'll give you the cards. And then you go, he loves me, and you have to say it all the time. He loves me to the bottom. And then he loves me not, throw away. And then he loves me. He loves me not. Then he loves me. He loves me not. He loves me. He loves me not. He loves me. He loves me not. And so your guy is, let's see. Oh! No! Okay, so where's all this going? Well, 100 years ago, when they made the Model T form, I'm not sure that they knew that Harry Gibson's car would look like that today. And in the same way, it's been 20 years since this serendipitous discovery. We have molecules that can do work, can rotate directionally, and even make other molecules. Where's this all going? I don't know, but there's lots of reasons to want to do nanotechnology. not least of which that Jessica Alba thinks that it's great. So, I hope I convince you it's possible to make new kinds of molecules, control the motion with them, and that we're really getting towards molecular things that you can consider as molecular machines. I haven't done any of this work. I'd like to thank all my group for their creativity and imagination and hard work. They're a really great lot from all over the world, as you can see with the flags. We've had some difficult politics in the UK over the last couple of months, and the real tragedy of Brexit is not the economic problems that it's going to cause the UK, or even the threat to peace and stability in Europe, but rather it's the loss of freedom of movement, which is so important, not just for science, but for all avenues of life. And the distressing thing is really the threat to the culture and the quality of having these sort of people that are so enriched my country for the last 40 years. But I don't want to end on a bad note. This is Edinburgh, which is a city I lived in for 10 years until very recently. This is called the New Town of Edinburgh, because it was all built between 1785 and 1830. And this is where we used to live here, in 13 Great Stewart Street. And if you walk down there into Ainsley Place, and then into Murray Place, down into Queen Street, and then into India Street, you get to 14 India Street. And 14 India Street is the birthplace of James Clerk Maxwell. And James Clerk Maxwell was, along with Isaac Newton and Albert Einstein, one of the three greatest scientists of all time, not because of Maxwell's demon or because of the discovery of the kinetic theory of heat, but it's because it was Maxwell that discovered that light was electromagnetic radiation. And that's a feat that Richard Feynman said 10,000 years from now, there can be little doubt that the most significant event of the 19th century will be judged as Maxwell's discovery of the laws of electrodynamics. And that's an astonishing thing to say, that the most important thing that happened in an entire century wasn't what a general did, or a president did, or other politician. It was what a scientist thought and what he gave to the world. And you can say the same not only for the 19th century, but for the 17th century, the 20th century. And it's great to see so many young people here today, because with you, the 21st century looks to be in great hands as well. So thanks very much indeed for having me. It's a real honour to talk to you today and to give this year's Kelly lecture. I'll leave you with one final thought, that sufficiently advanced technology is indistinguishable from magic. Thank you very much.
Thank you, David. I think we have a few minutes for questions. I'm sure you have many in this inspiring lecture. Luke.
So you have all these things driven by thermal motion, right? So your things are hopping back and forth, and then they come close to the barrier and push past the barrier, and then maybe get very close again. How much do you think about measuring rates of transport and diffusion, along chains and around the rotaxanes and all that?
Yeah, so we do that in order to work out the dynamics of the structure. So often NMR, in fact, is used to do that, to work out the kinetics of motion of the rings up and down the
So is that
the
just
chain.
changes in peak intensities or you have radiance NMR?
There's multiple ways to do it, but the simplest ones are where we have, say, this a thread that's got two different. You've got some system like this where you've got a thread with two different sites that the ring can sit on. Then you can measure the rate that it moves between them in various ways. So historically, the way of doing it would be make it very cold so that this is in slow exchange, and then find the temperature at where it becomes quickly. Nowadays, you can use pulse sequences to see how long these protons take to be in the environment where the ring is. So there's various different ways of doing that.
Yes? When you're constructing a polymer,
Yeah, so that's a great question. So there's many, many design faults in that initial system, and one of them is that one of the steps involves the transfer of the thioacetate, once the building block's been picked off, onto the growing chain. And as that growing chain gets longer, this cyclic transition state gets bigger and bigger and bigger, and so that becomes less and less likely that it will occur. So we've done theoretical studies that show that, or model studies, not experimental studies, but model studies that show that you should be able to make a 25-mer actually and get this cyclization to occur, but we've never made more than about 8 building blocks with these systems. There's some other disadvantages as well. The other problem is that it's not just the growing transition state, but the ring can access more and more and more of the track. So it means that it spends less and less time in the place where you actually want it, and then you've got to have this unlikely event of the cyclization occur. So everything gets slower and slower and slower the more building blocks you add with that sort of design. So that's another great point. So how can you try and overcome that? One way that we're looking at is, instead of picking up the building block and passing it always like this, so that it gets bigger and bigger and bigger, this transition state, we're looking at designing a system, or systems where we've designed it so that the ring, the arm picks up the blocking group and puts it on top of the next one, and then picks up, goes down, and that way the transition states are always the same size, and it's just that the chain is being moved, the growing chain is being moved along further. So if you could also, then that would get around the other problem. What we don't have in the current system is a ratchet mechanism to stop it going backwards, and DNA, the ribosome and DNA polymerases use ratchet mechanisms to get around that system, just like the ones that we developed with the Maxwells and rotaxane and the chemical fueled one. And so we're trying to apply those ratchet mechanisms as well to get around the other problem. So both very good points.
Just a follow-up, David, could you possibly have an insertion mechanism where the next site goes in close to the arm instead of at the end?
I'm sure that there are many, many different systems that can be designed. We haven't really thought of that other than that way of doing it. Other than that sounds reasonably similar to just we're inserting. If you're just picking up the building block and putting it under the growing chain on top of it, it's a little bit like an insertion. It's a little bit like it, but I'm sure that other people will develop other types of mechanisms.
One more question? Again, go ahead.
Have you thought at all about using molecular dynamics in designing your system? I know it's using mechanics right now, and you can't really model formation, but...
No, but molecular dynamics is certainly very useful for looking at the dynamics of these kinds of systems, for modelling the rates and how the things actually move. So my group use molecular modelling a lot in the designs, because making molecules, some of you will know, is an incredible amount of work, and I didn't cover anything to do with that at all. I just show these molecules miraculously appear on the slides, and it's not like that at all. And the biggest disaster is if you make a molecular machine and it doesn't work, because then you've done all that work and the design is wrong. So we try very hard through use of modelling to make sure that all the bits that we're designing work, and we make model systems before actually assembling the final ones. Having said all that, I'm always wary of being talked out of a design because the calculations say, oh, it's not right, and you've got to balance those sort of those two issues of wanting to have the best possible design and the most confidence in it without being talked out of something that might actually work because you've got something wrong with the modelling, or the modelling isn't accurate enough. So it's always a difficult judgment call.
Okay, let's thank David again for
I'm getting